The first speaker is Elena Fioravanzo from SIN. She's going to talk about the molecular modelling used for predictive toxicology within the AOP framework. This is really for liver toxicity with the LXR and PPAR gamma receptors. So this is 3D modelling. So this is stepping away from sort of work where you've got 2D structural alerts and trying to deal with receptor-mediated effects.
Thank you with this presentation through the to how molecular modeling can be used in the risk assessment framework as a predictive toxicology tool. And then we will go through what we have done within Cosmos to apply some of the molecular modeling methods in this framework. So we'll start giving you some background information, both on the AOP in relation to molecular modeling and above all on molecular modeling. Then we will see which of these methods have been used within Cosmos and the result that we got. There has been a shift in the last years between how in silico methods in risk assessment were thought, considered and used, and how they are considered today. And this shift is connected to the paradigm shift that you have seen before. Now toxicology would like to be based on the adverse outcome pathway. This means that the idea is to have a complete picture of how the adverse effect is caused. So to have a complete idea of all the biological events that are connected between the initiating event and the adverse, and the toxicity at the end. So within this new framework, the in silico methods are simply one of the tools that you can use and integrate with other tools. Well, this part here are just a list of methods that are available, and if in the past the in silico methods were considered only as a substitution of testing methods to save time and money, now they are simply a part of a wide array of tools that can be used together to have a complete picture of your compound, of the toxicity of your compound. So the idea is to use in silico methods in an integrated testing strategy approach. And I also put over there a new definition that has been recently introduced and has the same concept that was previously the ITS, and it is the integrated approaches to testing and assessment. And the concept is exactly the same, to use different tools all together, because different biological events can be studied and understood with different tools. So this is what you know is an adverse outcome pathway, and again I just wanted to highlight that it is simply, my vision is that it is simply a series of biological events that are connected to each other. And there is, initiating event, something that our compound is doing at the very beginning. And this initiating event is inducing a lot of our other biological events. So there is a cascade of events, and at the end you have your toxicity outcome, toxic outcome. Usually the adverse outcome pathways are described grouping these events connected to each other according to the level the level at which they are at the biological system. So they are grouped according if they are related only on the toxicant or the initiating event, the cellular level, organism, or after the population. Why we try to apply molecular modeling in this context? Because one of the initiating events can be an interaction between a ligand and the receptor. And some of the molecular modeling tools were designed to study exactly this type of interaction. Now we pass to some background about molecular modeling. I'm trying to be as general as possible because, well, I know that you have different backgrounds. So sorry for being maybe too naive for some of you. Anyway, molecular modeling has been used for more than 50, I think 60 years nowadays, in drug design to study the interaction between a ligand, if we are in risk assessment, we can call it a toxicant, and the macromolecule that is a receptor, or a toxicant and the target. And these interactions are studied. So the target is a macromolecule. Usually it has a binding site, so it's sort of cavity. And the ligand is enters this cavity and interacts with the macromolecule. We have a wide array of tools in molecular modeling that can be applied here, and we have some structural bioinformatics tools, such as the homology modeling or X-ray or NMR, to study the macromolecule structure. We have some structure-based tools and approaches to study the interaction between the two. I'm talking about docking, but we have also quantum mechanical methods if the interaction, if the reactivity is important for the interaction. Some of the drugs, they bind with a covalent bind to the receptor, and in that case, you can only use quantum mechanics to study those interactions. And also, we have mixed mode approaches that mix quantum mechanical and molecular mechanics. And we also have ligand-based approaches that are mainly used, that are useful when you don't know the target structure. So in this case, all the information that you have are from the ligand structures. You have different ligands that are active towards the same receptor, and you can, studying the structure of these ligands, find out why they are active and find out other structures that has the same activity. Within Cosmos, we use, and we will see later, docking method. So we selected receptors whose structure was known. So we didn't use the structural bioinformatics. The interactions were only non-binding interactions, so we didn't use quantum mechanical methods, but only docking methods. And we also used some ligand-based methods so that to have two different points of view on the same interaction. And we used pharmacophore analysis. This is just to remind you, when I'm talking about molecular modeling, which are the basis of molecular modeling? Well, molecular mechanics is the basis of the methods that I listed before that we have been using. These methods, they look at the molecule as being composed by points and bounds. And these points are the atoms. They have a dimension and they have a charge. They are charged and with a dimension. And molecular mechanics use the classical mechanics of interaction of physical points that are charged. So it is only that. So you know that if two points has the same charge, they don't like to stay very close to each other. If they have different charges, they like to stay very close to each other. And this is how molecular mechanics study the interaction between a molecule and the surrounding receptor. Well, it is more complicated than these, but these are the basics. And this is what docking makes. You have a receptor, you have different ligand structure. So with molecular mechanics, you can find which conformation of the single molecule can fit better the receptor, the site, the binding site, that is this cavity here. And when you have found all these, well, this was the animation, of course. These crosses were meant to come up later. Anyway, each of these can be fitted here. And with molecular mechanics, you can calculate simply which of these ligands is more happy to be here. So which can stay here. And so you delete the ones that are well, less stable as complexes, and you can calculate with the, well, you have a way to calculate the scoring for each of these, and the one with the best scoring is the one who is the molecule who is predicted to be more active towards this receptor. And this is, on the other hand, the ligand-based methods that we have applied. We use both a classical pharmacophore analysis, and here the basis are that, well, the definition of pharmacophore is a 3D configuration of structural features that are necessary to have that activity. So if you think that the activity is made by a molecule interacting with, well, the binding site, of course there are some points that are more important to have that activity. And if you have a group of ligands with the same activity, you can compare the structures of these ligands and find out, even if you don't know the receptor structures, which are the common points. And these common points are meant to be the essential points to have that activity. This is the classical pharmacophore analysis, and we use also a so-called e-pharmacophore analysis because we had the receptor structure. In this case, you find all the important features, analyzing only the ligands, and then you select the most important ones with the, well, considering also the receptor. And we use these different methods in parallel, so that to have a different scoring function, and then we use everything all together at the end. So when we were talking about molecular modeling, as I mentioned before, it was designed and thought and tuned above all for drug discovery. So when we started to think about using molecular modeling to study the interaction between a receptor and the toxicant, we wanted to highlight differences between drug discovery and risk assessment to be able to tune and to use to choose the proper parameters and to be able to apply a method that was developed and designed for drug discovery in a completely different framework. It's not completely different, but in a different framework. And we thought about the goals of the two, the differences in chemical space and the differences in the main task of in silico methods in drug discovery and risk assessment. In more details, well, the goal of the drug discovery is to design molecules. So the molecule is not given. The idea is to use, molecular modeling and docking as an example was designed to be able to give you ideas on how a molecule can be the most active towards that receptor that you want to interact with. If you think that the pharmacological effect can be described exactly as an AOP, you have an interaction at the beginning and you have the positive outcome at the end. So we are in the same structure. So, but the difference is that in drug discovery, you have to design a molecule, and this molecule has to be the most active as possible. In risk assessment, you want to know everything about your molecule, and you're not evaluating a single effect, but the whole toxicity of your compound. So it is slightly different. The other main difference is the chemical space. When we are talking about drugs, the chemical space is the drug chemical space that is well defined, because the drugs have to, well, they must have certain properties to be able to be absorbed by the body and to be able to pass all the biological membrane and to reach that point in your body that is where the receptor is. So it's not huge, the chemical space of drugs. When we're talking about risk assessment, we are talking about the chemical space of everything, and it is huge. It can include really very different and various type of molecules. So the chemical space is different. I included here, this is, I take it from a publication, and you can see here the red dots are registered drugs, and the purple ones are high production volume chemicals. And as you can see, the chemicals are here, so they are included in the chemical space of drugs. But the chemical space is really wider. And the parameters we are using to plot this chemical space are lipophilicity, well, the number of halogens that are included in the molecule, and this is the topological polar surface area. So really very basic parameters. And you see how, well, the chemicals are really, it is larger, much larger than the drugs chemicals. And this is the last point that we evaluated, the main task that in silico methods, how the in silico methods are applied within drug discovery and risk assessment. In drug discovery, the idea is to find the most active drug towards a given receptor. And so I would summarize the difference with this sentence, that usually in drug discovery, what you want to do is to minimize the false positive, because you want active molecule. On the other hand, in risk assessment, what you want is to minimize false negative, because you don't want to predict something as safe that it is not safe. So, you know, the aim, the goal, the task of the two in silico methods in these two frameworks are slightly different. The other thing is that usually in drug discovery, you are looking for strong interaction. In risk assessment, and here we were talking about repeated dose toxicity. Also, weak interaction towards a receptor can cause a toxicity at the end. This is the last difference. So we considered everything all together, and we selected the parameters to use molecular modeling applied in this different framework. And we used these studies as a case study to see how these parameters can be tuned. What we have done within Cosmos, we thought about this conceptual framework. So the first step is to select receptors that are relevant for an AOP. The other step was to review the existing knowledge about the receptors, about these receptors. And to review existing knowledge was also to understand how the structures were and if molecular modeling could be applied to these structures. Then we have carried out some molecular modeling studies. We developed a screening procedure using different molecular modeling approaches, and then we will see how these results can support the risk assessment. So we chose the receptor based on this AOP that was described within the SERA project, and it is related to liver steatosis, because the initiating event of this AOP is the activation, so the interaction with a receptor. And also, one of the gold compounds that were selected years ago within SERA, so this structure, gold compound means that these structures has been used within the different projects. So we could collect information about the same structures using different methods, and also what the other projects were doing. So we select this receptor. And then we also selected the PPAR gamma receptor. It is here. It is simply another AOP. You can see here that some of the events are related within these two AOP. And within Cosmos, it was also studied and proposed this AOP that starts from the interaction of the other receptor, PPAR gamma. This is why we have been using two different receptors in parallel, and both of them are related to liver steatosis. These are few information about the two receptors, and again what we have done mainly was to retrieve all the available 3D structures of all the receptors, to study those, to select the ones that were more relevant, and so on and so on. So the LXR receptor is a nuclear receptor, and it is involved in the metabolism of glucose and cholesterol. And the inhibitors, the ligands that interact with this receptor, can be grouped into two different types: the agonists that induce liver steatosis, and some others has an antagonist activity, and these are promising drugs for arteriosclerosis and other things. The binding site of agonist and antagonist is exactly the same. So the structures of agonist and antagonist are not very different. The differences are really small differences, but these differences induce a rearrangement of the structure of the receptor, so giving the different activity. What we did also was to collect and analyze known LXR binders to have a data set with which to validate the models that we were developing. And we collected more than 300 compounds with data related to LXR activity. And of course, they are mainly drugs, so they are mainly very active, but we also had some structures active or low active. And we did the same with PPAR gamma. PPAR gamma is also another nuclear receptor. It is involved in cardiovascular disease, diabetes, obesity. It has been studied in drug discovery to find something against obesity mainly. And it is part of a family with three subtypes of PPAR gamma, and they are structurally different. So PPAR gamma is a well-defined structure. And again, for PPAR gamma, different ligands were collected. We had more than 400 ligands with all the data to have a validation data set for the models. This is a summary of the different models, the different in silico methods that we developed considering these two receptors I'm here focusing only on the molecular modeling part because later you will see this afternoon there will be something related to the QSAR models and the structural lab. So I'm now talking only about the molecular modeling, but the results were found out considering all these models all together. This is the modeling approach that we use for LXR. I don't want to give you so much details. If you are interested, I can give you, well, there is a publication that is on review right now. Just to tell you that molecular modeling methods has to be applied in a different way for different receptors according to their structure. So we decided to apply this particular approach with LXR because of the initial study, and we applied a different approach to PPARy gamma. Just that. The methods are the same, they are applied in a different way. This is a summary. So we have done and we have seen the selection of receptors, high and which receptor we selected and why. The review of existing knowledge, you know that now we have collected all the available three-dimensional structures of the receptors and also information about the known ligands, known binders. We have done these modeling studies, and now we can see how the results and these methods have been combined together to get some results. So this is a case study that was submitted to SERA, and the idea in this case was to use molecular modeling methods to predict the binding the potential binding to and possibly the activation to these two nuclear receptors. We used two use cases. In one case, we had compounds. Well, the idea is to see if molecular modeling is able to predict if a compound is a binder of LXR or PPARy gamma. And in the other case, we had some known hepatotoxic compounds. And the idea is to see if molecular modeling is able to group compounds according to their possible AUP. So they are hepatotoxic, non-hepatotoxic, and maybe they are hepatotoxic for this AUP. So this is the first use case. We know nothing about these molecules. We can apply these methods, and if the molecule is predicted as binders of LXR or PPARy gamma, there may be some concern for steatosis. And these are the results that we got at the moment. So we had the known binders. We took the non-binders, we mixed with compounds with unknown activity, and we tried to find out if with other methods we were able to find out the known binders. And these are the results. How can you read this plot is that this dotted line here is the ideal. So ideally, the ideal docking would go in this direction. And this is the random. So if you choose randomly the molecules, you get these results. And this is the result that we got using different scoring function here. So you see, we are not so far from the ideal, and usually what it is done is to stop at this point. Well, the answer is yes. We are able to find the active known binders of LXR and PPARy gamma. This is the other approach that within these methods can be used. If we have molecules and we know that these molecules are hepatotoxic, if they are strong binders to these receptors, it may be that if the possible IOP for liver steatosis can be the interaction with LXR or PPARy gamma. And in this case, we use a data set that has been developed within the JRC, and it is composed by almost 100 compounds. They have been selected as hepatotoxic and with different AUP. And they are going to test these compounds to find out experimentally which is the AOP for each of these compounds. And this is something that is ongoing, so we don't have this experimental information at the moment, but we have run an array of tools on this data set. We had these, all the information. So now we are waiting for those results to see how our results were. I mean, if they were improving or not, the information about these hepatotoxic molecules. With PPARy gamma, what we have done was slightly different because we wanted to apply different approaches. So we have done the molecular modeling on PPARy gamma, and it was also done the substances with liver phenotypic effect were achieved from the O-repeat dose ToxDB that is within the Cosmos database, and this afternoon you will see what is the Cosmos database. And the chemotypes, ToxPrint chemotypes software was applied to find out which structural features are related to this type of effects. And these type of effects are related also to PPARy gamma interaction. So we mixed the two. We applied molecular modeling and this structural alert that was found with the ToxPrint, and we could find out. So this structure was found positive with both. It is the piperonyl butoxide, and it is in effect an hepatotoxic molecule and active towards PPARy gamma. And these are the conclusions. This is what we have done, and so we have shown that molecular modeling can be used with this approach within this framework.